Okay, now for today, we're going to look at this model for enzyme activity. Now, model basically means that we try to understand, okay, how enzyme actually functions by using some analogs. Means that something that we can to simulate, okay, the enzyme activity. So far we have two models to actually to show how substrate actually bind to the active site enzyme, and then the catalysis process actually take place. Okay, so what are these two models? So one we call lock and key model or lock and key hypothesis. Another one is induced fits hypothesis. So highlight these two. We're going to look at it in detail now. What is the lock and key model and what is induced fits model? So when you look at this lock and key model, is quite self-explanatory. In this case, lock and key, they must be complementary to each other. So in this case, lock and key is a model how enzyme catalyze the substrate reactions. And in this case, they must be exactly complementary. So when you look at the shape of the active site and the shape of the substrate, then you can see that they are exactly complementary. And in this analogs, okay, in this analogy, then you can see that, okay, the lock is the enzyme, the key is the substrate. In this case, are you clear? The lock represents the enzyme, the key represents the substrate. So it means that you cannot use simply any key to open a lock, correct or not? So they must be complementary to each other. So what is the active site? So active site basically means that the keyhole represents the active site. So it means that only correct key, the substrate fits into the correct hole of the lock. So it means that the substrate have to bind to the active site. So it means that the shape of substrate must be complementary to the shape of the active site, the enzymes. Are you clear? Okay. So in this model, we have to know that when a substrate molecule collides with an enzyme whose active site shape is complementary, the substrate will fix into the active site and enzyme substrate complex can be formed. How they form this enzyme substrate complex? So they form what we call the temporary bond. So for example, they form hydrogen bond. They can form the ionic bond. Okay, and according to this model, it is possible for enzyme to catalyze the reverse reaction. Now, this is very important, okay? Because why? The active site, if you look at the diagram here, you look at the shape of active site and the shapes of the substrate, so they are complementary. But when you look at the shape of product and the shape of enzyme, active sites, they are complementary. So means that in this model, we actually think that it is possible for enzyme to catalyze the reverse reaction. So it means that formation of the bond, okay, and also the reverse process. It is possible because why? Exactly complementary. Are you clear? Okay, but this model is not widely accepted model. Initially, yes, but now we realize that not really accurate model. Now, why we say so? Look at this. If this is the lock, okay, this is the keyhole, correct or not? And then you put the key in. Okay, you put the key into this the lock. So the key actually is a substrate. The lock represents the enzyme. Now, what will happen here? Look at this. When you slot in the key and you turn, the key remains as a key. Your key won't break. Can I see that? But enzyme, what will happen to the enzyme? Your lock will open. Cannot. So means that this model actually is not accurate because we do know that enzymes shouldn't change the shape. The enzyme shouldn't undergo any net change of the chemical geometry or chemical shape, okay, or structure after the catalysis process take place. So when you put in the key and then you turn, and then the lock open. So means that key remain as the same. You don't break the key. The key won't change the shape. But what change the shape here is the enzyme. So there's a lock and key model only can explain how this substrate can bind to the active site perfectly because the shape, their shape actually complementary to each other. So that's why you can put in the key, but they cannot explain why the catalysis process actually take place. Are you clear? Okay, so this is a lock and key model. So after this model, then scientists come out with another model called induced fit hypothesis, or they call it a handshake, shake hand, right? Shake hands hypothesis. Okay, handshake hypothesis also. So this is actually is a widely accepted one. Induced fit model is currently a more recent model, which is widely accepted as describing the way enzyme work. So in this case, you can see that the shape of active site highlight not exactly complementary. Okay, can you see that? Not exactly complementary, but change shape in the presence of specific substrate to become complementary. So means that this substrate, when approaching this active site, the shape of active site actually can change and make it actually more complementary. So means that substrate molecule must be a correct molecule. If it's a wrong type of molecule, it won't induce the change of the shape. So in this case, understand that the active site actually flexible. They are flexible. So means that it's not a rigid, you can change the shape, but only slightly. Are you clear? Okay, so that the substrate can fix it. Okay, so the enzyme change shape slightly due to interaction between the substrate chemical groups and also the chemical group of the site change R group amino acid. So this shape change make the active site fits even more tightly around the substrate, cause the stressing and distortions of the chemical bond. So as I said that, guys, if this is the active site, and then you want to put this substrate in, so definitely you have to change the shape of the active site, clear or not? But the thing here is when you change the shape, the active site like this, so means that the protein, the enzyme want to return back to original structure, so they exert the force. Okay, they exert the force onto, okay, this substrate, distort the chemical bonds in the substrate. So that's why we say that decrease the energy needed to break the bond. So decrease the activation's energy. So induced fit model actually a better versions to tell us that how enzyme work. First, they are not totally exactly complementary, but with the correct substrate, you can actually induce the change in the active site shape so that now the substrate actually can bind. So when the substrate can bind like this, then because the enzyme want to return back, it's something like spring, right? So you pull the spring and release the spring want to return. So same goes to this, okay? So something like this, okay, the shape change already. So it exerts the pressure or the force onto the substrate molecule. So making this substrate molecule now, okay, or stressing the substrate molecule distorts the chemical bonds of the substrate. So that therefore you can see that they require less energy to break the bonds. Okay, so this induced fit model also, induced fit hypothesis, also known as the handshake model. When you shake hand, try to imagine, guys, when you shake hand, what will happen? Okay, so now let me on my camera here. Let me show you guys something. So you can see me, right? Okay, now handshake model, let's say you want to shake hand, right? So first what you do, you open your palm like this, correct? You can see that. I want to shake hand. Can I see that? So one is enzyme, one is the substrate. So according to this model, be careful, guys. Even though in our syllabus, we always say that the one that change the shape is active site, but our substrate also can change the shape. Okay, remember. So it's something like you shake hand, right? Can you see that one? My hand, you can see, right? Another hand. So first you shake hand, you can see that it's not complementary, right? But once you approach each other, what you do? Can you see that? You start to hold the hand. So this is something like enzyme do. You can see me, right, guys? Can see my hand or not? Yes or no? Okay, can you see that? Okay, good. Now look at this. So what I call it handshake model, handshake model. So you shake hand, right? Can you see that? So first you don't do like this. Okay, you cannot, right? Can you see that? You cannot. Can you see that? So but now you will see that this is handshake model. So this is my hand, this is your hand, for example. So we come close to each other, then we change the shape. So make it complementary. Can you see that? Complementary. Okay. So how you break the bond? You try to imagine now you shake hand with the young kids and see 2 years old, 3 years old, and then you exert the pressure, then you can break the bone. So it's similar to the enzyme active sites that actually now tightly, okay, fix with the substrate and then exert the pressure. Can you see that? Exert the pressure. So when I exert the pressure, can you see that the bond actually distort? So with this distortion of the bond here will allow the activations of the energy. So that's why induced fit model explain. Right? Induced fit model actually explains the process of the catalysis compared to the lock and key model. So induced fit model currently is a more acceptable model, okay, compared to the lock and key, because lock and key cannot explain how the enzyme actually catalyze the reactions and how enzyme actually lower down the activation energy. But induced fit hypothesis, yes, they can actually tell us that because of the change of the shape, right? It's something like the rubber band. When you pull the rubber band and you release the rubber band, it return back. Correct or not? So same goes to the enzyme active site. They are flexible so that they can actually tight, a means of fits even tightly around the substrate, provided that substrate must be a correct substrate. If the substrate is not a correct substrate, then useless. They would induce. Okay, so it means that in terms of induced fit model, we wouldn't be able to catalyze the reverse reaction. Now, can you see that? Because first, you must have correct substrate. Only these two can induce the change in the shape of the active site. So if you look at the product now, product is not a substrate, so therefore they cannot induce the change in the shape of active sites. So with this, we know that now enzymes won't catalyze the reverse reaction unless I say unless the products actually can induce. Okay, unless, but generally we can say that it won't actually catalyze the reverse reactions. Okay, so that's all for the parts of the model. Do you guys have any questions? Feel free to ask now.